So we learned about synaptic plasticity and we saw the classical NMDA receptor dependent mechanism that you can find in textbooks. We learned about STDP, and we've also now learned about intrinsic plasticity, which appears to have a lot of the same hallmarks, which is to say it's, NMDA receptor dependent and also probably calcium dependent. Um, is there anything else? Well, the final thing I want to touch on is this figure that we saw when we introduced LTD, right? So when you stimulate weakly, you get LTD. As you stimulate stronger and stronger, you go through no synaptic plasticity, and then eventually you get LTP. And I've said, if you get low frequency firing or high frequency firing, but who is to say what low and high frequency firing is, right? Like, I'm sure there must be some cell in the brain where 1 hertz firing 1 times a second is unusually fast, On the other hand, there are cells out there where firing at, what is this, 10, 20, 30, 40, 50 hertz is not that crazy. It's regular business, right? So what do those cells do? Are they just inducing LTP or LTD all the time? What if the threshold, this point here, it's usually called theta M, um, where you go from LTP to LTD, what if that was variable depending on your recent history of activity? I.e., if you've been highly active, then this shifts to the right, saying that we need to be even more active to get LTP, while we can be moderately active and get LTD. What if you're a cell that never fires action potentials? Well, then this whole curve should shift to the left, saying, look, if you can even fire a couple of action potentials, we'll start talking about LTP. And if you fire, once a year, then we'll start talking about LTD. And this is what they have seen. In fact, um, yeah. Anyway, so here, these are the kind of theoretical curves that you're expecting, where you get LTD with low frequency, if the low previous activity, or you need very, very high input to get LTP if you've had high previous activity. And we've seen this in theoretical studies. So this is from the visual cortex of mice that have been dark red and light red, so red in the dark, so their visual cortex is basically inactive. And in those, you need relatively low input to get LTP, while in light red normal animals you need higher frequency input to get LTP. And in fact, we can mimic this using in the brain slice. So if we apply some priming stimuli, so we just make this input be active, not strong enough to induce LTP or LTD, but just active enough. We make this whole pathway active, and then at some other pathway to the same neurons, we then supply the high frequency tetanus. What we see is that we get less of an LTP. So the point is that we've had a lot of input. Historically, this neuron is starting to realize, oh, I think I'm in a highly active pathway, so it gets less LTP. And if we do the same thing, we actually get more LTD, right? Because all of a sudden getting this very low frequency input is highly unusual, and the neuron says, okay, I'm going to experience LTD. So we can change this threshold between when you go from LTD to LTP depending on the history of the cell. So I've really only just touched on synaptic plasticity and intrinsic plasticity, right? There's all sorts of induction possibilities. There's all sorts of responses. As I said, there's non-NMDA receptor dependent LTP. There's non-Hebbian, so you don't need the postsynaptic cell to fire. There's anti-Hebbian, which is this STDP, but with the other way around. There's homosynaptic and heterosynaptic LTP, right? So only the cells that are involved in the pathway in the tetanus get stronger, while other times other synapses can get stronger too, depending on how you're doing it and depending on what cell type you're working in. That said, to me, the proof that the NMDA receptor is involved in memory is incontrovertible, right? So no matter what kind of crazy synaptic plasticity mechanism you favor, if it doesn't involve the NMDA receptor, I've got a problem with it. But I should tell you that there are some crazy ideas out there, at least to somebody who was brought up, learning about LTP, like I mentioned here, particles of RNA being passed from presynaptic to postsynaptic cell to presynaptic cell. I didn't even touch on presynaptic mechanisms of LTP and whether or not they truly exist. Um, so this is a huge field, and in the 90s, this was the coolest area to work in. So there's a lot to talk about. So I've really just touched on it so that you can at least follow along with some of the ideas about synaptic plasticity.